All right, so the lab today, it's still thinking about the urinary system, but from a very different perspective. And basic idea is urinalysis, which, a lot of times people think, pee in a cup to, see it's a person on drugs or something, but it's really a common way to just see very basic functioning of the body. And there's some more typical things you test for. There's lots of specialized things you can test for. We're going to go over some of the major things that you test for and kind of how you do it. So, let's first talk about the idea of a clean catch. Clean catch or a midstream catch. So the basic idea here is when you start peeing, there could be all sorts of contaminants on the entrance to your urethra and stuff. So what you want to do is I have to make it yellow. You start the flow and assuming that that's going to kind of clean things out. So you don't want the initial part of when you start peeing. Instead, you let it go, let it kind of clean things out, and then midstream catch. Then you put the thing there to catch it and get the urine to analyze. That way you're less likely to have contaminants that are going to cause artifacts in your data here. So that's what clean catch means, or midstream. When we do our lab next week, where we actually care about the total volume of urine that you're doing, then obviously it's not a clean catch. There you have to catch every last drop if you want to know the total volume you peed out. But if you're just interested in, what are the qualities of your urine, what's in there, what's dissolved in there, you just need a nice little sample here. Things that you check, before you do any of the fancy chemical testing, you can just check the color. Obviously color is going to depend on a variety of things. It can have a lot to do with how concentrated it is. If you are more dehydrated and you are conserving a lot of your water, that means whatever you're excreting is going to be more concentrated and less volume. It tends to be more darkly colored, tends to smell more intensely as well. Color, though, can also be from vitamins or stuff you're eating. Like you can have, if you've ever taken these B vitamins, you get this like fluorescent pee, or beets and other things can give you reddish pee. It can also be color because, blood or something. So you do want to pay attention to the color. People who do this for a living have all sorts of words, what is it? I think straw or amber or blah, blah, blah. There's lots of classic words for how light or dark. You check, is it cloudy? It shouldn't be cloudy. That's usually a sign that there's some kind of infection, if there's like lots of bacteria in there or something. So before you even look at this other stuff, you kind of check some of these big pictures. But then you're interested in some of the more chemical constituents and things like pH and things like that. And in the old days, you actually had these complicated things, like you'd have to take a bit of urine, mix it with a particular little crushed up pill and look for the blah, blah, blah. It was this big to do just to look just for glucose or just for ketones or just for, then for pH, you use your little pH paper, and everything was its own test. And What we have now are these things called multi-sticks. And it's basically this little, I mean, I'm drawing it bigger than it is here. It's about like 3 inches long. It has all these little squares on it that measure different things, and you just dip it into your urine, and then you pull it out and you wait like, some between 30 and 60 seconds. And each of the squares is going to give you a readout on some other property of your urine. It'll tell you about the glucose, or whether or not there's blood present, or what is the pH, or things like that. I should mention, before we go into the details of the multi-sticks, I need to mention specific gravity, since that's one of the things we're going to be testing. So specific gravity. This is a typical thing that you assess in the urine. This is basically the ratio of the density of your urine compared to the density of water. So it's a ratio. And what is density?
How tightly packed the molecule is.
So it's, and what would be the units of density?
Grams over milliliter?
Yeah, it could be grams over mils, or, yeah, so it's basically going to be mass, some mass divided by volume. Right. So I could have a certain volume of styrofoam, and it's going to have way less mass than that same volume of lead. Lead has a much higher density than styrofoam. This is a ratio. This is actually one of the very few numbers in our class that is not going to have units, because the top and the bottom are the same units, it cancels out. So it's a pure number. It's just a ratio. If you've just been drinking tons of water and your body is getting rid of lots of water and your urine is super dilute and it's almost like peeing out water, what's the specific gravity going to be?
It's lowered.
Well, give me a number.
0, 1.
Which one? 0. Remember, density of your urine over the density of water. If the density of your urine is pretty much like water, what would be the value of this ratio?
Be 1.
1. So if your specific gravity is 1, that's basically like water, right? That means that you are peeing out this super dilute urine and there's not much dissolved in it. The more solute that you're peeing out, the more stuff that is dissolved in the water, this number is going to go up because the density of the urine is higher than the water. Typical values are like 1.01 or 1.02. By the time you're getting to like 1.04, this is like pretty high. This is pretty concentrated. I can think ahead. Hold on. So just to give you a sense of what these numbers mean, like if somebody has a specific gravity of 1.040, they're probably pretty dehydrated because their body is holding on to a lot of water and the density of their urine is very high. So this would be considered high. this would be considered low. And this would be considered almost like water. This is like water. So that's specific gravity. For the multi-sticks, it actually is just going to be able to be easy to read with a color. Next week, we'll look at another way to measure specific gravity using a little urinometer, a little float. But for today, we just think about it like this. So now, I think we're good to go here. So, urinalysis lab. So these are the things that the multi-sticks can detect. So I'm going to just briefly run you through them so you have a sense of what we can be looking for and what it means, because part of the lab is interpreting the results. You're going to do these tests, but then you have to interpret like what is likely going on with the patient. So glucose. One of the things you can look for is glucose. Should glucose be found in your urine?
No.
No, typically your body is going to take it all back. That tubular reabsorption brings it all back. If there is glucose, it's usually a sign of some problem. It could be you've just totally sugar binged and you've overloaded your body with so much sugar before it's had a chance to adjust and you're spilling over some. But typically, it's considered a sign that you have diabetes mellitus. This is the type of diabetes where you have a dysregulation of your, how you regulate glucose. I should probably, let me put this as a, diabetes. There are actually different things with the same name. There's diabetes insipidus. This is something else. This is, you lose, so P way too much. So this is where your kidneys are having trouble actually retaining water. So diabetes insipidus, and it's insipidus means it doesn't have a flavor, right? Insipid is tasteless. It's because the urine is not doesn't have a flavor because it's basically like water. It's super dilute. As opposed to the one that we're talking about now is diabetes mellitus. Mellitus means sweet because the urine usually has sugar in it, so it tastes sweet. This is, I think, way before there were multi-sticks. Somebody could taste the urine a little bit, and if it was sweet, sugary, that would be a sign that there is of diabetes happening. There are two different types of diabetes mellitus. There's type 1 and type 2. Type 1, this is more of an autoimmune one where your body attacks the little islets of Langerhans in your pancreas and you don't make insulin. We'll talk about insulin in more detail later, but basically insulin tells your cells that there is sugar available in the bloodstream and they should take the sugar up from your bloodstream. So if you eat a candy bar or slug down a glass of orange juice, your body will release insulin. The insulin is a hormone that tells your cells sugar, and the cells take it up, and then the sugar levels in the blood go back down. If you do not make insulin, then the sugar levels just get higher and higher in the blood because The cells aren't getting the message to take it out. Type 2, you actually are making insulin, but your cells are not responding to the insulin. So this one is a little different. You do have insulin, but your body is not responding to it. This one here, this is an autoimmune one. This one's often called also juvenile onset. This one's often called adult onset. This one typically happens later. This is often associated with obesity. When people are more overweight, in fact, often just by losing weight, this will resolve. Although, again, because of juvenile obesity, it's more common for kids to actually get type 2 diabetes because they're obese, which is kind of a shame. But in both cases, you have the same result. Either you're not making insulin or the cells aren't responding to the insulin. Glucose levels in the blood go up, which means glucose in the filtrate is much higher, which means you're overwhelming the capacity of the PCT to reclaim it all, which means you have glucose in your urine. So that's why it's one of the big things that you worry about if you see glucose in the urine, that there is some problem here. Okie dokie. Bilirubin. Bilirubin I mentioned briefly as one of the products that's involved with the recycling of the heme group from the red blood cells. Typically, it just gets dumped into your, as part of your bile. Again, it's breakdown products from the bilirubin are what make your poop brown. But if you have liver problems, it can get spilled into your blood, and it then ends up in the urine, and it's like in hepatitis, you get bilirubin spilling out. Anything that kind of messes with your liver can causes leakage of bilirubin. So you want to be aware if that's showing up or not. Ketones. I mentioned ketones a long time ago in class. Ketones show up as intermediates when you're breaking down fats for energy. So if ketones show up, that means your body is breaking down fats for energy. It could just be you're fasting, so you need to break down fats for energy. Or it could be you're on the caveman diet. You're just not eating any bread, so all you're eating is fats and proteins or something. Or, it could be something else. Often if you have diabetes, you get a mix. There's going to be glucose showing up because your cells aren't absorbing glucose. And you have ketones because since the cells aren't absorbing glucose, they're having to go into lipid breakdown instead. So glucose along with ketones together is another sign that you're likely in diabetes. But ketones, again, can just mean you're fasting or just your diet isn't giving you carbohydrates, right? It's not necessarily means you're a disease or something. Specific gravity we just talked about. Blood. Should there be blood showing up in your urine? What is most likely scenario? So especially like when we do this in this class, we often, it's not uncommon that somebody has blood in their urine. What is that probably from?
A UTI?
So it can be from a UTI. If there's blood, it can be from a UTI.
Probably your liver's not working.
It's often contamination from menstrual blood. If it's a female of reproductive age, it's often, more often than not, it's from that. Like there's a saying, and like in medicine, what do they say? Like when you hear hoofbeats, think horses, not zebras. Like, all right, so what's most likely? Like, oh, there's blood in the urine. This could mean all sorts of stuff, but make sure, like, rule out the more likely kind of boring common thing first. That it's just some contamination there. It can also be like you've just been in some big bar fight and got some major kidney shot, and just bleeding, who knows. In general, it's not a good thing, but again, often it's actually just from contamination. pH, what should the pH of your urine be?
Like, or the same pH as your blood?
No, not necessarily.
A little more acidic.
Right, so this is where it's important to remember the function of the kidney, the pH balance. Right, so if your kidneys need, maybe I'll go. Here's your kidneys, here's your urine, here's your body. If your body pH is going up, I might have to spill out more bicarbonates. If your body pH is too low, I might have to spit out more H pluses. Right, so the kidneys are going to do whatever they need to do to maintain pH balance. And it's going to depend on what the pH of your body is, which depends a lot on what kind of stuff you've been eating and stuff. Like a typical diet, your urine tends to be slightly acidic. I think more vegetarian diets, often you end up having more of an alkaline urine. But the urine, there's no correct answer for what the pH of your urine is. The pH of the urine depends on what adjustments do your kidneys need to do in terms of excreting bicarbonates or hydrogen ions in order to keep your pH balanced. So urine is going to depend, the pH of the urine depends on your diet and all that. All right, let's continue here. Protein should not show up. Big proteins should not make it through that filtration membrane. When they do, it's usually there's some problem. Urobilinogen, let's not worry too much about it. It's also indicative of liver disease and all that. These two we'll talk about, the nitrites and the leukocytes. Both of these are things that are associated with urinary tract infections. So the nitrites are something, they're a metabolic byproduct of a lot of the bacteria that cause UTIs. So if the nitrites are showing up, that means likely you've got these gram-negative bacteria that are living up in your urinary tract. Leukocytes, again, those are the white blood cells, which we've been talking about as part of your immune system, which is also a sign that there's some UTI going on if they show up. So these are all the things that you're going to be looking for. This little video I'm going to show you now, this is just kind of showing what it looks like when you do the little multi-sticks. Yeah. Yeah, I mean, in general, urine is, you don't have to be as paranoid about urine as you do with blood. Blood is definitely something you got to be super careful about. Urine tends to be a little more sterile. Obviously, this guy doesn't want to get pee on him, but this is, yeah. Right. Again, this is what I was talking about. The color does tell you stuff. You know, normally if you're, it should be kind of a light yellow, straw colored. If it's very dark, that means it's probably really concentrated due to dehydration, although it could be from other things in your diet. If it's got reddish or pinkish, it could be blood, which is not a good thing. Brown, like I was talking about those bile pigments, like the bilirubin and stuff. This guy is intense. And again, cloudy, often from bacteria. So this is actually important. Like when these things get old, they start just turning colors without anything and they don't work properly. So we actually end up throwing these out every once in a while because we realize like they've started going bad on their own. And again, this was a clean catch. Like James here started peeing and then put the little cup under the stream once the flow had started. So, you know, the initial peeing flushed out and hopefully cleaned things out. So there's different amounts of time, like 30 seconds, you can check glucose. So in this square here, again, if it stays blue, that means negative. If there was glucose present in the urine, it would start turning green or brown, depending on how much. And you know, the color will, you know, you can't, it's kind of hard to see here, but it actually gives you a quantitative measure, like how much glucose, depending on what color this thing turns. You know, bilirubin is also after 30 seconds. Ketones after 40 seconds, specific gravity after 45 seconds, other ones after 60 seconds. You know, again, we can see a specific gravity is around here. It looks fine. No blood because this square would have green on it if he had blood in his urine. pH, it's kind of acidic. It's kind of more down here around 6 because it's kind of this brighter orange. So you just compare the color on the dipstick to the color on this key, and that lets you know what the value is for each of these different things we've been talking about. This guy is...
To complete the procedure, I would document the results of the urinalysis in the patient notes.
All right, so that is Mr. kind of no fun. This one here, this has a groovier accent. She's talking more.
Remove the lid from the urine sample after ensuring it is mixed. Remove a urine test strip from the container, ensuring that you do not contaminate any of the test patches. Insert the test strip fully into the urine sample and tap off the excess. Note the time and hold the test strip against the result scale on the container. Read off the results at the appropriate times by comparing the colour of the test patches against the scale on the container. Record your results.
So yeah, she's...
Remove the lid from the urine sample.
So I want to just show you both of those so you just have a sense of what the actual procedure is. Again, you know, when we're back at school, if you ever want to come into lab and do this on your own pee, you know, I'm happy to hook you up with some of these test strips if you don't have access to them. You know, here is the graph I have just here in the unit. I have it printed out. I also have a JPEG that you can open up if you want. So, whoop. So you're going to need this to do this lab, because the lab is going to ask you to evaluate some strips that you're going to see. So that's this first thing. The other thing is urine sediment. One of the things we're going to do, what we would have normally done is, in fact, let me just kind of describe this briefly as well. If you take a little test tube of your urine, you know, it just looks kind of clear. If, you know, assuming nothing's wrong with it, it looks clear. But the reality is it does have little things floating in it. It has little cells that got washed off from the walls of your urethra. It has little crystals. It can have these things called casts, these little weird protein things that take on the shape of things within your kidney tubules. So what we do is we take this test tube with your urine, and if we have, assuming there's tiny little things in here, and we want to concentrate them and pull them all down to the bottom, what are we going to do?
Centrifuge.
Exactly, we're going to centrifuge. And what you're going to end up with is some little pellet of stuff. And then what you do is you just, you pour out most of this, and then you resuspend this. So this is what we call the sediment. The sediment is this dissolved stuff in your urine. And it's another thing that is useful clinically to try to understand if you're healthy or not. So what you can do is then take this and look at it under the microscope. We call it sediment analysis. There's a variety of things that you can see. Here, in fact, let me go back to the screen. So over here, view single page. This all here, these are basically little crystals and stuff. This is what you might see underneath the microscope when you look at this stuff that you've concentrated by putting your urine under, through the centrifuge. You can see little crystals, these little uric acid crystals, these more amorphous little urates. It's really common to just see all this kind of stuff in there. These are really tiny, right? If crystals got big, then you've got kidney stones, but these are itsy bitsy things that you only see under the microscope. You know, there are other ones that are only found when you have diseases. So by looking at this stuff, you know, this is kind of showing there's different kinds of crystals. You can find cells. It's actually really common, super common to just find like epithelial cells just from your urethra. It could find white blood cells if you've got a urinary tract infection. These weird things, these things called casts, they are, again, there's this protein, it's the Tams horsefall protein that kind of gels inside the kidney tubules. It takes on the shape of things inside the tubules. So by looking at these little casts, these little moles of coming out, it can tell you if there's some problem inside your in your kidneys. So, you know, for this class, I don't want you are not responsible for recognizing any of these or understanding really what it means, but you should know they exist. You should know that sediment analysis is part of urinalysis. You should understand that if you take your urine and you spin it down, you're going to see stuff, and that stuff gives you information about somebody's health. You know, what I would ask you to do, you know, I'm not going to have you run through it now because we have other places you can do it on your own. You know, but
the complete urinalysis.
You know, you can watch this. This is he's a veterinarian, but he's talking about just some of the stuff you find in urinalysis, in the sediment analysis.
Indicates the presence of inflammation, which could be infectious or non-infectious. And
right, so watch it. It's 10 minutes. You know, watch it. And what I've asked you to do in your in the write-up here, you know, I said, in addition, in your write-up, mention at least 2 different things that may be found in examining the urine sediment and what they might indicate. So when you're writing it up, I want you to spend a little bit of time getting a sense of what kind of things you find and what they mean. So please, in your write-up, and again, you can find that both through the lab manual and also through the little video. And if you search on YouTube, there's lots more stuff on sediment analysis. This part 1, evaluate your own urine. You know, I'm saying here, obviously we can't evaluate our own urine because you don't have these multi-sticks at home. Or I'm assuming you don't. So what I have here, I've kind of split it up depending on your last name. If you're A through G, you're going to do patients A and B. Each of these, you know, if your last name begins with H through L, you know, if it's M through O, you know, so I give you, there's 2 patients depending on which your last name is. And each of these, I'll just open up patient A. You know, it is a multi-stick. And what you need to do is evaluate what's going on there. Whoops, hold on, I've got a battery emergency. I think we're back. You know, again, just what would you say, just from the very first thing we're looking at, if you're looking here, what is this telling us?
There's a lot of glucose.
A lot of glucose on this person. So what you're going to do in your lab notebook, you write down each of there's 9 categories, 1, 2, 3, 4, 5, 6, 7, 8, 10 categories. And for each of the categories, you are going to assess, you know, what this multi-sticks tells you about that subject, you know, and then say, what does this mean? Does it just look normal? Does something off? If it's off, what might be happening? So that is what you're going to do for part 1. For part 1, you are going to pick 2 patients based on this little chart here. For those 2 patients, you are going to just evaluate the multi-sticks results that are simulated in the little PDF and write down the results and then write down what it means. So that makes sense? And then I said, in addition, spend a little bit of time thinking about sediment analysis and describe at least 2 different types of sediment and what they might mean. So that is part 1. Part 2 is similar, except instead of looking at things through a PDF, you're going to watch Becky. So there's 2 more patients.
Okay, this video is going to cover part 2 of the urinalysis lab, evaluate the unknown samples. So here we have 2 urine samples.
So these are different than the A and B above. And before you get the data, you want to write a chart in your lab book already, like glucose, bilirubin stuff. So you're ready. So as she's going, I'll just, rather than sit through it all, I can just show you fast. Let's do it fast so you can just see it.
From patient A and patient B, we have the multi-stick test strips. We have a timer, and we have a blotting tap, paper towel. So let's go ahead. Let's test patient A's urine. So we're going to be putting the multi-stick in, just like we did in part 1. Tap it, blot it, start the timer, and our first reading will be in 30 seconds.
Nice.
Okay, 30 seconds. So we're reading the test strip. So we're measuring glucose, bilirubin, ketones, specific gravity, blood.
Right. So use this video of somebody actually doing it for real to actually record the data. This is just kind of want you to get a sense of what it really feels like to do this. And again, you can just see, you can see what things look like on the strip. You can see what things look like on the key. And in your lab notebook, you're gonna, normally we do this with 2 people because of this very reason. Normally the one person is holding this thing with urine on it, figuring out the color, and another person is writing things down in their lab book because you can't, it's kind of hard to read it and write it and not get urine over everything. So for part 2 is just a little more of the same. Basically, patients A and patients B, write down what their results are and what you think might be up with them. So it's pretty straightforward. Any questions about that? Okay. So that is this urinalysis lab. I mean, I've talked a bunch to give you background, but the actual doing it is going to be pretty straightforward. It's basically going to be evaluating 2 patients for part 1, just through the pictures, and then 2 patients for part 2 in looking at the video. The other thing I want to talk about is the lab for next week. This is one where you actually do have a objective and a hypothesis that you should come up with ahead of time. So I want to just spend a little bit of time describing that, what the lab is about, just so you have a sense of what we are up to. And it's 11, so we've got plenty of time here. I'm going to do this, and then we can also, if people have questions about things, about the exam, we can talk about that as well. So next week, so this is the lab that's happening next week. And the basic idea is we are going to vary a variety of things. So we have our independent variables and we're going to have our dependent variables. So independent variables, we're going to have one thing. In fact, what would be an interesting thing to vary if we're looking at how the kidneys regulate things?
Water.
Water. So we're going to integrate amount of water that people drink, amount of H2O intake. So that's one thing. We can have some people drink a whole bunch of water. Some people barely drink any water at all. That's going to be one of our independent variables. What's another thing we could vary? That would be interesting.
Salt intake
Salt.
or electrolytes.
Amount of salt intake. We're going to have some people eat salt tablets and some people not. So some people will take in excess salt, some people won't take in excess salt. So these two seem pretty obvious. So the other one we added in, just because there is this kind of basic thought in most of the world, like, that caffeine makes you want to pee. We're also going to have caffeine intake as an independent variable. So we're going to have some people take in a bunch of caffeine and some people not. This is going to be through a pill, because otherwise we have if they were drinking coffee, then there's a confounding variable of they're actually getting lots of water with their caffeine. So for the caffeine intake, they're just going to pop like a Vivarin or a no-dose. So you're basically getting the pure caffeine. So these are our independent variables. And the dependent variables, we're going to be examining the urine. And what are interesting things that are going to tell us, be interesting to look at?
The amount.
So one is going to be the volume. How much are you peeing?
The color.
So color could be interesting, but that would be a little trickier. We're gonna do sodium chloride concentration.
The frequency.
For the frequency, we're actually gonna force people to pee every half an hour, so we can get the data across many different subjects. We're gonna look at the volume. We're gonna look at the sodium chloride concentration. And then that other thing we were talking about, the specific gravity. Basically how concentrated it is or not, just everything. In general, you're going to expect these two to be pretty similar. If you've got a lot more sodium chloride in your urine, you're probably going to have a higher specific gravity, right? Because specific gravity is just a measure of dissolved stuff in your urine. So this is the basic idea here. Independent variable, we're varying the amount of water intake, salt intake, caffeine intake, and then we're seeing how that affects the volume, sodium chloride, and specific gravity of your urine output.
So there have to be, sorry to interrupt you, Professor, there have to be 3 hypotheses of each independent variable.
It's
For
a little more complicated than that. So I'm going to go to the actual lab itself here now. So here's the lab. there are 4 groups that you can see here. Group 1, Group 2, Group 3, and Group 4. Group 1 is drinking lots of water and no coffee, no caffeine, or no sodium chloride. Group 2 is taking a lot of water and also taking in salt. Group 3 is taking in almost no water. 80 milliliters is basically an itsy bitsy cup. It's what you need to swallow the salt tablet and not much more. Group 4 is drinking a lot of water and also taking caffeine. Again, this is just in your lab manual. You don't have to write this down. So in your lab manual for next week, though, in your, for objective, what would be the objective for this lab based on what I just said?
How does water intake, salt intake, or caffeine intake affect the volume, salt concentration, specific gravity of the urine?
Exactly. Did everyone hear Laura? Like that was just in a nutshell, the perfect objective. And so what you want to do then is use each of these 4 groups. And for each group, you want to make a prediction for our dependent variables. For each group, you want to talk about what do you expect the volume, salt concentration, and specific gravity to be like. And it's going to have to be relative, right? We don't have any ability to have an actual volume or concentration. You're going to have to be group 1 compared to the other groups, or group 2 compared to the other groups, group 3 compared to the other groups, group 4 compared to the other groups. And when you're thinking about your hypotheses, you really want to just think about the idea of balance. If somebody is drinking a lot of water, what is the kidney going to have to do to maintain homeostasis, your water balance?
Increase filtration rate so you can pee more.
It's going to increase urine production, right? So you're going to lose more water to keep the water balance. If you're taking more water in, you got to let go of more water to keep your balance. What about if you are barely drinking any water at all? What is probably going to happen to maintain water balance?
Reduce urine output.
You're going to reduce your urine output. You're going to conserve water, right? So when you're thinking about your hypotheses, I want you to think like, don't get too caught up in details. Think about these big picture of the kidneys working to preserve balance. Same thing with salt. If I'm not eating any salt versus if I am eating salt, how am I going to expect that going to be changing my urine output? Or change.
If you eat a lot of salt, you're going to pee out a lot of salt.
Yeah, if I take more sodium in, I'm probably going to get rid of more sodium, right? Because to maintain balance. To be fair, this amount of salt is not that much. We used to do this with more salt, but then people would start feeling really uncomfortable and stuff. So if I lowered it lower and lower, this would be like eating a big bag of pretzels or something. It's not like a crazy amount of salt. What else? Caffeine. Caffeine is always funny because people always think they know what caffeine is going to do, but it'll be interesting. So you have to use your, you can do a little Googling. You can, figure out what do you think is going to happen, the effect of caffeine on these different things of urine output. I should also, what else should I mention? No, that's about it. What I want you to do, before, I know that you've got other stuff because you're studying for your exam, but minimally, I would like you to come up with, again, the objective, which is very straightforward. Lauren just kind of came up with that there. And then come up with a hypothesis for each group. What do you expect their relative volumes, sodium chloride concentrations, caffeine, or specific gravities to look like? And again, you should have a why, right? I expect the volume of this group to be, among the highest because, or this will lowest because, or this will be somewhere in between because. Make sure your hypotheses have a reason. And I did include what's going on here? I want to get rid of this. I did include, actually, you can see this is Becky. If you want to see what it looks like, this is Becky talking about how the setup looks.
We're going to be covering the kidney regulation of osmolarity lab today. And in this lab, we have students sign up for one of 4 groups. Group 1 is to consume 800 milligrams of water and nothing else. I'm sorry, milliliters, thank you, milliliters of water. And this cup with this red line is 800 milliliters of water. And so we want those students in group 1 to drink that as quickly as they can as the start of the experiment. Group 2 is going to also drink 800 milliliters of water and consume 1.5 grams of salt. And here we have salt tablets. Each tablet is 1 gram. So those students would take 1.5 tablets, and they should swallow them and not chew them. And they have plenty of water to swallow them with.
All right, so what happens? So what happens if you just swallowed a bunch of salt granules and just all of a sudden inside of your stomach was really super salty all at once? Anybody? If your stomach was full of salt water, what's going to happen?
You're going to vomit.
Why is that?
Because water is going to rush in to the stomach because of osmosis, and then you're just going to have to puke to get rid of all of it.
Exactly. So what's going to happen is osmosis, all the water rushes into your stomach gets all distended and stretching out, and the next thing you know, you're hurling. These salt tablets are designed to dissolve kind of slowly so you can absorb it as it's getting released. It's why if you're in a lifeboat on the ocean, you can't just drink seawater to stay alive. If you try drinking seawater, you will just hurl, because seawater, your body's water comes in and it's all going out.
Group 3 is going to ingest 80 milliliters, and the red line shows 80 milliliters of water, and the same amount of salt, 1.5 grams, so 1.5 tablets of swallowing the tablets, not chewing the tablets. And in group 4, they're going to consume 800 milliliters of water and 1 caffeine tablet, which is 200 milligrams of caffeine. Plugging vibrating. So each group should ingest that as fast as possible, as comfortable as possible. And then that'll be times 0. And then after 30 minutes, students will empty their bladder in various containers using the restroom. And then they are...
Again, it's not going to be a clean catch. This is going to be every last drop. so you can actually get the total volume.
Are going to take various analysis on their samples. They're going to measure their volume, and we have different sizes of graduated cylinders to hopefully match closely what the overall volume of urine that was collected every 30 minutes. Specific gravity, we'll show that in just a second. And also sodium chloride concentration, we'll show that next.
So in the basic kind of way the experiment will happen is that, T equals 0, you intake your water, your sodium chloride, your caffeine. And then we'll do measurements. We'll measure volume, sodium chloride concentration, specific gravity, at T equals 30 minutes, T equals 60 minutes, T equals 90 minutes, T equals 120 minutes. So we'll be able to see over time how these dependent variables are being affected by this initial intake of water, salt, and caffeine, and how the kidney responds over the next 2 hours to maintain homeostasis. So are there any questions about this? So in the, oh yeah?
We need to have a total of 12 hypotheses then.
Yeah, or I could say it's like 4 hypotheses, and each of the 4 hypotheses has 3 subparts. Something like that. I would like you to scan and submit this pre-lab, before we actually do the lab on. It's going to be after we're done with our exam. We're going to actually be looking at this, but I would like you to have thought about this and kind of written up this initial thing before we actually do it. So this is a submit PDF of kidney regulation of osmolarity lab pre-lab, just like we've done with other ones.